Hello, everyone, and thank you for the organizers for inviting me. It's really fun to be here, and so we're going to switch gears a little bit, go from print into the world of software. I'm going to tell you a little bit about this web-based platform for genomic data exploration that I designed for the New York Genome Center. And to make it fit in here, of course, we used lots of visualization tools. So, but first I want to talk a little bit about what it's like working as a designer in a software context. And that's where this sort of cyclical nature of design that you're probably all familiar with to varying degrees really comes full circle, because something like a piece of software that's out there that has users, that doesn't just get published once and then people look at it and they never come back. You hope they come back time and time again and they keep using it and actually use it for their work. So that's where you really get a sense of how repetitive this design process can be. Where you're building something, you're trying it out, you're evaluating it, and when you put the user in the whole context, you find out very quickly what's working and what's not working, because you get feedback from people. And so essentially, that's what user-centered design is taking people into consideration at every one of these steps. So before you even start designing, you do some interviews with users, with potential users, or people that are similar to who you think might be using it. Then you just start very loosely. You don't need to get too hung up on details right away. And figure out, do we even have a concept, a process that seems promising, that works? And you can get feedback right away from a user, even on these sketches. Sort of like, one of the diagrams that Lucy showed earlier in her talk, that was kind of like a wireframe of just laying out the elements of everything that needs to be on screen without really talking about style or substance, but really just the placement and the broad concepts of what you want to show. And you review that design with users, and then as it's getting more refined, you get to a point where now you can start making it interactive, and you write some code to try out, how does that really work? And so I did that, interviewed, because this is about genomics, interviewed the people that work in the labs that generate all the data and subsequently analyze it. Not all of them, but some of them. Most of the people that analyze it work on a different floor where it's just all the computational folks that deal with all this biology that got turned into digital data now. And so the job for our tool was basically to take that mess of data that's really has very little meaningful information in the individual data bytes, and turning it into something that a biologist can look at and make sense of it without having a whole staff of computational people on their side to help them. So actually, before I get started on this, let me get a quick show of hands. Who here works in biology in some context? All right, so that's about a third maybe. Who is involved in any kind of software engineering? All right, small group. And who are writers or people that deal with words mostly? Even fewer. Okay. So the rest of you are all medical illustrators? Okay, great. So I don't need to go too deep into it, but I'm still going to talk about what is really at the heart of this all, DNA. That's what the genome is made up out of. And we subdivide it into genes, and then in those genes you can identify genomic variants. So what does DNA really look like? I mean, we know we have about a 2-meter long string of it curled up into every nucleus in every single one of our cells. And so when you simplify it in an abstract way, you can just show it as a line. And then if you zoom in, you would see the sequence of letters that really makes up that line. So 4 nucleotides that get repeated, 3 billion letters altogether. And then to make it a little bit more manageable, we've divided it up into chromosomes. And then if you look more closely at the chromosomes, we finally get to the genes. So those are areas on each chromosome, and they have funny names like RBM8A or EGFR and TP53. Those are all abbreviations for actual functions of the proteins that they encode. So RNA binding motif 8A, epidermal growth factor receptor, I believe, and tumor protein 53. So when we look at genomics, the interesting part is to find all the things that are different in people. So not just everybody has a TP53 gene, but not everybody has the same mutations in a TP53 gene. So that's how you can find out what makes you different from other people, or what makes your cancer different from other cancers when you're looking at disease. And so you identify these variants, and they can be labeled with a coordinate in the chromosome, and then you indicate what's the change. So there's an A that changed to a C, for example. So essentially, that's a lot of what genome sequencing gives you in the end. For each individual, you get a list of about 3 to 4 million spots in your genome where you differ from everybody else. But recently people have been looking at this for a long time, but recently we started adding to MetroNome, to this exploration platform, another level that goes beyond the genomic variation to look at what really makes different tissues differently. So we have one genome. It's in every one of our 30 to 40 trillion cells. But somehow our body manages to make 78 different organs from that same information. And there's 200 different cell types. So how does that really work? And the answer to that question is it's gene expression. It's how many genes are turned on and how many genes are turned off in a particular cell type or for a particular organ. And all that regulation basically tells our body how to behave like a heart cell, or how the skin cell behaves like a skin cell, and so on. So we tried to see, okay, how can we add that to MetroNome? And we got a little bit of help. I don't know if you remember the Ice Bucket Challenge. This was a huge success in crowdfunding for a disease that's been very, very poorly understood, ALS. And so we got some of that money together with one of the scientists from the New York Genome Center that is very active in the ALS consortium to help them build some tools to look at that. And so ALS is a neurodegenerative disease that basically affects your motor neurons and makes them die. Stephen Hawking is a very famous example, but he's also very unique in the way that he lived a very long life, with ALS. Typically, people die within a couple of years of being diagnosed. So you don't have much time once you get that diagnosis in a lot of cases. And so they're asking us, okay, we have all these post-mortem samples that we're getting, and we're looking at the nervous system in a fairly detailed fashion. So they gave me this list of 13 different areas that they're tracking, where they got samples from. And they can measure the gene expression in each of those areas separately. So I said, okay, let's try and map that onto a diagram where we can then show a heat map, sort of for each gene. How is, for example, the SOD1 gene, that's one that's already known to be related with ALS. How is that expressed in all these different areas? And so I started out with this composite that I made from a bunch of images I found on the internet. And they said, okay, we don't really care about the vertebrae that much. Oh, and by the way, we also got liver and we got some cell line samples. Well, turned out later they didn't really get that much data from liver and cell lines, so we ignored that. But what they told me is, oh, and by the way, we want to be able to distinguish between left and right hemisphere. So that's an interesting problem. So I can't use that side view because where's left, where's right? So I started thinking about that and looked at brain images from all these different angles, and don't you know it, there's like all these occlusion problems. Like there's parts you can see from the top that you can't see from the side, and things you can see from underneath that, I mean, as illustrators in the medical field, you're all familiar with those kinds of issues. So I looked at, okay, how do they do that in games and things like that? They have these maps that let you draw a face and an entire head in a two-dimensional plane. It's sort of like a world map that we're used to looking at, where the whole ball is sort of rolled up into one flat piece. And they did that for the head. So I started mapping out all these different areas onto that 2D head map. And while you can see everything, it becomes pretty obvious that it's not really intuitive. So it takes a lot of effort to sort of wrap your head around it. And so we said, okay, maybe it's better to just forget about the realistic anatomy. Let's just make it a diagram somehow, something more schematic. And this way you can stack all these different layers up on top of each other. You sort of roughly put them in the place where they go in the head and how they relate to each other. But now we can have one view where you can divide it into left and right and see all those different areas. So the cerebellum that's normally hidden underneath the head becomes visible, and all these other issues, like the motor cortex subdivisions that they have there that are really difficult to see in an anatomically correct brain. They, now just become areas that we have there. And so the first thing I heard back from them was, what's this mouse brain that we're supposed to work with here? Because they said, oh, it looks like a mouse. It's so smooth, it's so elongated, And they're used to working with images from that, from mouse models, so damn, okay. All right, so I figured, okay, let's try and fit that more into like a skull outline, and that's a little bit better. But they still weren't quite happy with it until finally I made it a little more organic looking, right? So we have some of the folds in the outer area, all the dividing lines are a little more organic looking, and okay, now they were finally happy with it. And so here now you can see, how according to the expression levels, different areas of the brain are colored differently and of the spinal cord. And so that then sort of formed the basis for turning this into a panel inside of our application, where you can now see a little more. The labels went away because what happens with software, you have interactivity. So you can have labels on demand when somebody mouses over something. You know, here, actually, well, the label shows up down here, but it tells you what it is that you're looking at. And it tells you here where that individual lies. And in the background, you have a little bit of data visualization to show you in a histogram what the rest of the cohort looked like. So this person there is right in the average. That's where everybody is, apparently, for that brain region. And then we have a diverging scale here at the bottom that shows a C-score that's calculated on the fly, you know, from whatever the search result was for your query in that system. Is this above average or below average, basically? And then we have a few other things here. You can switch to a different gene, for example, with a drop-down menu. And then we realized, okay, there's all this other data also that they've been collecting besides the expression data. We have somebody measured the pH value of that brain sample. And they have a whole range of them there. Because they were interested to see, does that really have any correlation with gene expression or a certain disease outcome or something like that. Then they also have something called the RNA integrity number. That's basically a quality score for how much RNA you get out of a sample. And so there's a whole range of values there. And then there's the last thing is the post-mortem interval, meaning how long was it since that person died until they took the sample, until the autopsy was performed. And so it's looking pretty good. A lot of them are less than 20 hours after death, so that's why they got a lot of good data. But then somebody was saying, okay, so now I can select a range here on this histogram. How come when I select the entire range, I'm getting suddenly fewer samples than I had before, you know, even though I selected the entire range? Well, it's because of this column over here. The samples where the values were missing for that measurement. So that's another thing with data visualization that's always really important, is that uncertainty factor in some cases, or even to just account for measurements that were not taken. Because if you leave that out of the picture, then it distorts all your numbers, and you never get to any sort of meaningful statistics. All right, so here's what this looks like now in the whole context of MetroNome. So there's a lot going on here. We basically have two different queries. I don't know if you can still read that in the room, but basically I was selecting a bunch of genes here. So we're selecting a cohort of people that have mutations in those genes. And I filtered it by subject group and used the non-neurological controls over here. And then over on the blue side, we have ALS spectrum disease. So it's basically, you know, healthy people versus sick people. And down at the bottom here, there's a gene diagram for this DYNC1H1 gene. And you can sort of see a little bit of a difference between the healthy ones and the sick ones, in the sense that there are a few more variants over here. You have all that summarized up here also. So there's 793 variants in the healthy controls versus 1,216 in the people with ALS. And the two diagrams here sort of show a difference in how that gene is expressed. So in the healthy group, we have more red here, meaning higher expression values in the frontal cortex and then the occipital cortex and the cerebellum. Whereas here, the occipital cortex is a blue shade, meaning it's really underexpressed compared to the overall average. And yeah, so that's how it goes. Then you can select subgroups from that, and it makes the difference more pronounced. But just to come back to the beginning and to wrap it up, after we did all this, we get back to the cyclical nature of design, because now they just published this huge amount of data from the GTEx consortium, which was a global effort of scientists collecting expression data from 50 to 60 tissues, and they also did a lot of brain sampling. And so suddenly it turns out, oh, the most important thing is really we want to compare our samples to the GTEx samples. So we need a map that works for both. And in GTEx, they didn't care about left and right hemisphere. So we're probably going to go back to something that looks a little bit more like an anatomically correct brain. But yeah, that's how it goes. So thank you very much. And thanks everybody from the MetroNome team that worked with me on this. I think one of my software engineers, Sylvester, is over here. But yeah, thank you all.